Hello and welcome back to the HIVRNAtestguide.com YouTube channel, your trusted source for HIV testing with over 4,500 HIV testing labs across the United States. For more information, check the link in the description of this video or the bio section of our channel.
Welcome back to the Deep Dive. This is the show where we really pull apart the most complex research papers, we find the core insights, and make sure that you walk away well-informed and maybe just a little bit surprised.
And today we are tackling one of the greatest, truly one of the most enduring challenges in global health.
The quest for a functional cure for HIV.
It's a mission that you really can't overstate. When you look at the numbers, the sheer scale of this disease it's staggering. Since 1981, HIV/AIDS has claimed 44 million lives.
44 million.
And even with all the incredible medical progress we've made, UNAIDS still reported something like 630,000 AIDS-related deaths globally in just 2024.
And that context is so crucial because I think for a lot of people listening, the story of HIV research kind of ends with the success of antiretroviral therapy, ART.
And it is a monumental success. You can't deny that.
Oh, absolutely. ART maintains undetectable viral loads. It's transformed HIV from, a death sentence into a manageable chronic condition for millions of people. I think the number was nearly 40 million worldwide as of late 2023.
But that very success creates a whole new set of challenges, Challenges that researchers are now desperate to overcome.
Right, because RT isn't the end of the story.
Not at all. Daily oral ART presents these huge logistical and medical hurdles. You need strict, lifelong adherence. We still see side effects, potential drug interactions, and because the virus mutates so quickly, drug-resistant variants can emerge really fast if that regimen is ever compromised.
And these logistical headaches, I imagine they're magnified in high prevalence areas, right? Like across sub-Saharan Africa.
Exactly, and that is precisely why the global medical community is so intensely focused on finding therapies that are either long-acting, so dosing maybe once or twice a year, or the ultimate goal, a complete cure.
So what's the goalpost for that? What does a successful cure actually look like from a scientific perspective?
Well, the community has a very clear optimal target product profile. First, obviously, it has to be safe and tolerable. But specifically, it must keep HIV RNA levels below the transmission threshold. That's 200 copies per milliliter. And it has to do that for a long time, at least 3 years, ideally forever, without daily pills.
And it has to work for almost everyone.
That's the other critical piece. It needs to be effective in over 90% of individuals with a really minimal risk of drug resistance popping up.
That is an incredibly high bar. And when researchers talk about a cure, they're usually talking about two different concepts, aren't they?
That's right, yeah. First, there's the sterilizing cure. This is the complete, permanent elimination of all HIV from the body. Total eradication.
And that's only happened a handful of times, right? With incredibly risky procedures.
Exactly. A few times using high-risk stem cell transplants, which are just not scalable for the general population. So the much more practical goal is the functional cure.
Long-term remission.
Right. The virus is still there, maybe as a non-replicating provirus, but it's permanently suppressed without needing any treatment. And that's where today's deep dive is really focused.
We're looking at a new generation of incredibly powerful tools. Broadly neutralizing antibodies, or BNAbs, and how researchers are combining them with other things to try and achieve that long-term remission.
But to understand why we even need these new tools, we first have to really grapple with the core problem, the fundamental reason HIV is so hard to cure.
The latent reservoir.
The latent reservoir. Even when RT crushes the circulating virus down to undetectable levels, HIV establishes this reservoir of infected cells, and it does it incredibly early in the infection.
It's like the virus's perfect survival mechanism.
It is. The HIV genetic material, it integrates itself into our own genome, into our DNA. It becomes a provirus. And in a lot of these infected cells, that provirus just becomes transcriptionally silent. It goes latent.
It's an invisibility cloak.
A perfect one. Because the cell isn't actively making viral proteins, the immune system has no signal, no alarm bell to tell it that the cell is infected.
So the killer cells, the cytotoxic T cells and NK cells, they just fly right by.
They're completely blind to it. So they're hidden, protected from any immunological attack. RT stops new infections from happening, but it can't touch this hidden cache. So if you stop treatment,
the virus comes roaring back.
Usually very, very quickly. And it's not just the cells that are a problem, it's also where they're hiding. We have these anatomical challenges called sanctuary sites.
Like the brain, the genital tract, gut tissues.
All of these places. And the issue in these sites isn't always latency. Sometimes it's just about drug penetration.
So the drugs can't get there in high enough concentrations.
Precisely. Some antiretroviral drugs, just because of their chemical properties or blood flow to that area, they might not fully penetrate these sites. You get these pockets of suboptimal drug concentrations.
Which could allow for slow, low-level replication, a constant seed for the reservoir.
Exactly. The consensus isn't definitive on whether that's continuous active replication, but the fact that you can have these pockets where drug levels are lower, it just adds a huge layer of complexity to achieving a true functional cure.
And if eradicating the reservoir is hard, just measuring it seems almost impossible. How do you even know how big the problem is? The sources we looked at highlighted 3 big challenges with the assays, the tests they use.
This is a massive, massive bottleneck in cure research. The traditional gold standard, the quantitative viral outgrowth assay, or QVOA, it actually underestimates the reservoir size.
Underestimates? How?
It only gives you an estimate of the minimum frequency of cells that can be sort of forced to replicate in a lab dish. And it's incredibly laborious and expensive to do.
Okay, so then you have the proviral DNA PCR test, which sounds like it would solve that, but it goes too far the other way.
Way too far. It greatly overestimates the size because it can't tell the difference between a defective provirus, which is basically junk DNA that can't replicate anymore, and the actual replication-competent virus that's the real threat.
So it counts all the duds.
All of them, and up to 90% of what it counts might be defective, just noise.
So to fix that, they developed the intact proviral DNA assay, the IPDA.
Right, and the IPDA is a huge technological leap. It's a very sensitive digital droplet PCR assay, and it's specifically designed to infer which proviruses are intact, the ones that don't have those common defects.
So it filters out most of that noise?
A huge amount of it, yes. But even the IPDA isn't perfect. We've seen studies that show after many years on RT, even some of these intact proviruses can become permanently silenced by epigenetic changes. So they're not a threat, but they still show up on the test as potentially infectious.
So you have all these imperfect tests, which means the only definitive way to know if a new treatment has actually worked is to have the patient stop their medication. An analytical treatment interruption, or ATI.
Which is inherently risky.
It sounds incredibly risky. And what was astonishing reading the sources was the variability in how these ATIs are done.
You've hit on a major ethical and scientific tension here. When a person living with HIV stops RT, they risk several things. Opportunistic diseases coming back, the reservoir expanding, immune exhaustion, and of course, potential transmission.
So how can one trial restart RCAR on only after the viral load hits 500,000 copies, while another one restarts at just 1,000? That's a massive difference.
It's a very complex risk-benefit calculation. It really depends on the treatment being tested. On one hand, if you set the bar too low, say at 1,000 copies, you might stop the trial too early. You might miss the chance to see if the person becomes a post-rebound controller.
Meaning their own immune system kicks in after seeing a little bit of virus and then takes over control.
Exactly. You're stopping the immune system from getting that boost, from learning its lesson. You might be censoring a potential success story.
But on the other hand,
if you let the virus get up to 500,000 copies, you're prolonging that viremia. That massively increases the risk of reservoir expansion, immune exhaustion, and, most importantly, transmission to partners. It's a very fine line to walk.
So the stakes are incredibly high when you're testing any potential cure.
Extremely high.
Which brings us to the exciting part, the tools themselves, specifically broadly neutralizing antibodies, or BNAbs. So let's break this down. What exactly is a BNAb, and what makes it broad?
A BNAb is a very special kind of antibody. HIV's surface is covered in this sugar coating that's constantly changing, constantly mutating, which lets it evade most of our immune response.
Right, it's a moving target.
But BNAbs target the conserved regions of the HIV envelope protein, the parts that are crucial for the virus's basic function, the parts that can't really change without breaking itself.
So they hit the virus where it can't mutate?
Precisely. They hit these specific vulnerable sites, like the CD4 binding site, the V1, V2 loop. These are the core machinery of the virus. And because these sites are conserved across many different strains, the antibody has breadth. It can neutralize a wide variety of HIV variants.
And the technology to find these has gotten way better.
Oh, dramatically better. The first generation BNAbs, they just didn't have enough potency or breadth. Resistant strains would emerge really quickly. But the newest generation, developed using single-cell cloning, shows a 10 to 100-fold increase in potency and a 2-fold increase in breadth. They're just fundamentally better weapons.
And they don't just block the virus, right? They're designed to rally the body's own defenses.
Yes, they have this dual functionality. First, the arms of the antibody, the Fab fragment, binds directly to the virus and prevents it from entering a host cell. That's direct neutralization.
That's the passive defense.
Correct. But then the tail of the antibody, the FEE fragment, actively recruits the rest of the immune system. It flags the virus for destruction by other immune cells, a process called opsonization. It triggers something called the complement cascade, which can directly rupture infected cells.
And it triggers ADCC, antibody-dependent cellular cytotoxicity.
Which is crucial for clearing the reservoir. The FC fragment binds to an infected cell that's expressing viral proteins, and it acts like a beacon, calling in killer cells, like CD8 plus T cells or natural killer cells, to come and destroy that infected cell.
But there's a third effect that seems to be the real key to a long-term cure, not just temporary suppression, the vaccinal effect.
The vaccinal effect is the theory that the BNAb infusion isn't just a passive drug. It's actively teaching the body's own immune system how to fight better.
It's like a training exercise for the immune system.
That's a great way to put it. The idea is that these antibody virus complexes that form, they get processed by our immune cells in a way that leads to a much stronger and more durable T cell response, specifically from those potent CD8 plus T cells.
So the BNAb flags the enemy and tells the immune system, memorize this guy.
Exactly. And this isn't just a theory. It was strongly supported in non-human primate studies. When they took monkeys who had achieved viral suppression with BNAbs and then depleted their CD8 plus T cells.
The virus came back.
Instantly, in every single animal. It proved that those T cells were absolutely key to maintaining the control that the BNAb had initiated.
So you need the BNAbs to start the process, but researchers are also engineering them to be better, to last longer in the body.
Absolutely. One of the biggest breakthroughs has been extending the half-life. They've created what are called LS variants by modifying just 2 amino acids in that FC tail region.
And how does changing just 2 tiny pieces of the antibody make it last so much longer?
It's a really clever bit of engineering. The modification increases the antibody's binding affinity to a receptor called FcRn. Now, this receptor is basically the body's recycling system for antibodies. It grabs them and pulls them back into the bloodstream instead of letting them get degraded.
So by making the antibody stickier to the recycling receptor?
It just stays in circulation much, much longer. This extends the time to viral rebound from weeks to potentially months. And that's a huge logistical win, especially in places where daily medication or frequent clinic visits are a major hurdle.
It's not just a better drug, it's a more practical drug.
Exactly. And beyond lifespan, they're getting creative with the structure. We're seeing bispecific antibodies that target 2 sites at once, even trispecifics.
Have those worked well?
It's been mixed. There was one trispecific antibody, SARA 441236, that was safe, but its antiviral activity was pretty limited. Seems the individual parts just weren't potent enough when they were all stuck together.
And then there are nanobodies. The name alone sounds futuristic.
They're fascinating. They are tiny, single-domain antibodies that come from camelids, so camels, llamas.
The future of HIV treatment comes from llamas.
It might. Their antibody structure is much smaller and simpler, which means they can sometimes get into little nooks and crannies on the virus that conventional, bulkier antibodies can't reach. It just shows the incredible diversity of the approaches researchers are trying.
That sets the stage perfectly for the big discovery in our sources. The antibody called O4A06. This seems to be a real step change.
It is a remarkable discovery. It came from a German research group, and what they did was screen blood samples from 32 people who are elite neutralizers.
These are the rare individuals whose bodies naturally produce a very powerful antibody response to HIV.
Exactly. They were hunting for the blueprints of natural resistance. They tested over 800 different antibodies. And this one, O4A06, which came from a woman in Tanzania, just immediately stood out.
How potent are we talking here?
We are talking about unprecedented potency and breadth. In the lab, it neutralized 98.5% of the nearly 340 different HIV variants they tested it against.
Wow.
And that included many variants that are known to be resistant to other powerful BNAbs. One of the leading virologists, Alexandra Tricola, said that O4A06 alone achieves an efficacy you usually only see when you combine multiple different antibodies.
That's a game changer. What's so special about its structure that it can do that?
Well, it targets the conserved CD4 binding site, which is a common target. But the key is a unique structural feature, an 11 amino acid insertion. It acts like an extra finger.
an extra finger.
Yeah, that's how it's been described. This extra little loop reaches deep into a conserved crevice on the virus, grips on incredibly tightly, and makes the whole bond super stable.
Yeah.
They ran computer simulations and found that no single mutation could really allow the virus to escape that grip.
So it's locking on to a part of the virus that is just too critical for the virus to change.
It's a true Achilles heel. And importantly, it also seems to have that vaccinal effect we talked about. The lead researcher, Florian Klein, noted that viruses blocked by O4A06 were also better recognized and actively eliminated by the body's immune system.
So it's got the lockdown and the immune recruitment, and what did the animal data show?
In humanized mice, a single shot of O4A06 completely suppressed the virus. No rebound for over 12 weeks. And that's a crucial finding, because with older antibodies, you'd often see resistance emerge, and the virus would come back within days or weeks. Sustained control like this is very, very promising.
And that suggests it could be used for prevention, too.
Absolutely. Based on models from previous large prevention trials, the researchers estimate that a single injection of O4A6 every 6 months could provide over 93% protection. That puts it right up there with modern long-acting PrEP drugs.
This all sounds incredible. So what's the catch? What are the caveats?
Well, experts like Dr. Christoph Spinner are rightly cautious. He pointed out that this is all preclinical data. It's from the lab and from mice. You can't just directly transfer that to real-world effectiveness in humans.
Right.
We still need rigorous clinical trials to figure out dosage, tolerance, and how it holds up against the true global diversity of the virus.
But the work is moving forward.
It is. The antibody has been licensed to Vera Biotechnology, and clinical trials are being prepared. The signs are, as Alexander Tricola said, definitely promising.
Okay, so even with a potential superstar like O4A06, the big lesson from all BNAB research so far is that you can't rely on just one agent. It's all about combination strategies.
Combination is absolutely key. It's the same principle as RT, which uses multiple drugs to block viral escape routes. So researchers are now strategically combining BNABs with 3 other types of agents: immune modulators, latency reversing agents, and therapeutic vaccines.
Let's start with the immune modulators. This is part of that shock and kill idea, right?
That's the premise. Immune modulators like toll-like receptor or TLR agonists work by flipping a master switch in our cells called the NF-κB pathway.
Okay, what does that master switch do?
NF-κB is the central regulator of the cell's immune and inflammatory response. When you flip it on, it does 2 critical things. First, it boosts the whole antiviral immune response, more T cells, more NK cells. Second, and this is the key for the shock, it reactivates the latent HIV provirus, forcing it out of silence.
So the modulator provides the shock, and the BNAB is there to handle the kill. But the early clinical trials showed that sometimes you get the complete opposite of what you expect.
And that was one of the most important and surprising lessons from this whole area of research. Take the Titan trial. It combined a TLR9 agonist with 2 powerful BNABs, and the results were really disappointing.
It was antagonistic. The combination was worse than the BNABs alone. How does that even happen?
It suggests we still have a lot to learn. In that trial, they found a significantly faster decline in the BNAB concentration in the blood of the group that got the combination. So the time of viral rebound was actually shorter, 9.5 weeks, compared to 12.5 weeks for the group that only got the BNABs.
The immune booster made the treatment worse.
It did. The researchers had a couple of theories. Maybe there was a direct drug interaction. Or maybe the timing was all wrong. Giving the agents before the ATI might have limited the amount of virus available to really prime the immune system, so the response just fizzled out.
But another trial with a different immune modulator, BEAT2, had more success.
It did. The B2 trial used type I interferon with BNABs, and they saw much better viral control. 86% of participants kept the virus suppressed for at least 26 weeks, and 40% never even had to restart their ART after the 38-week study period.
That sounds like a functional cure for 40% of them.
For durable viral control, it's excellent. But when they looked deeper at the reservoir itself, the results were less clear. The combination didn't actually decrease the size of the reservoir, and it didn't seem to increase the long-term T-cell responses. So it was controlling the virus, but maybe not clearing the source.
Which leads us to the second strategy, combining BNABs with latency reversing agents, or LRAs, a more direct shock.
Exactly. LRAs are chemicals designed to do one thing: force the provirus out of its silent state. The main ones are called histone deacylase inhibitors, or HDAC drugs like romidepsin.
And they work by changing how the DNA is packaged.
Right. When the virus is latent, the DNA is wrapped up really tightly around proteins called histones. HDAC inhibitors prevent that, forcing the DNA into an open, active state. This forces the cell to start producing viral proteins, essentially unmasking itself to the immune system.
And the eClear and Roadmap trials, which both use romidepsin and a bNAb, give us this incredible contrast, a perfect case study.
They provide a profound lesson about the importance of antigen. So the eClear trial gave the treatment to people who were ART naive, meaning they were recently infected and had a lot of virus circulating.
And in that group,
It worked. 80% of them maintained control for at least 12 weeks off RT. The combination boosted their specific anti-HIV T-cell immunity.
but then the Roadmap trial used the exact same drugs, the exact same schedule in people who had been on ART for a long time.
And it showed no benefit at all. These people were chronically suppressed, so they had very little circulating antigen. In that trial, the combination was no better than the bNAb alone in delaying rebound.
So the takeaway is that timing is everything. You can shock the virus out, but if the immune system isn't already primed and ready to go,
The kill part fails. You need that recent exposure to antigen to keep the specific CD8 plus T cells on high alert. If you don't have enough kill potential ready, then shocking the virus just speeds up the rebound.
it all suggests you need an immune system that is constantly being trained. Which brings us to the third strategy, bNAbs plus therapeutic vaccines.
This is the most ambitious goal. The idea is to replicate the kind of robust, long-lasting immune response we see naturally in elite controllers, which is mainly driven by those strong CD8 plus T cells.
So the vaccine provides the long-term training, and the bNAb provides the immediate protection.
That's the idea. And they're using every kind of vaccine platform you can imagine. DNA vaccines, viral vectors like adenovirus and MVA, new mRNA vaccines, and traditional subunit vaccines.
And combining 3 or even 4 things, a vaccine, a bNAb, and immune modulator, sounds incredibly complex.
The complexity seems to be necessary. In a primate trial, a triple combination of a TLR7 agonist, a bNAb, and a vaccine outperformed all the dual therapies. 70% of the animals ended up controlling the virus on their own.
And are we seeing that translate into human trials?
We're seeing early promising signs. There's a trial, NCT04357821, that combined a DNM, a VA vaccine, a TLR9 agonist, and 2 bNAbs. 70% of the participants got at least partial viral control. And critically, one person in that trial has maintained complete viral control for over 18 months off ART.
A year and a half, that is the definition of a durable functional cure.
It absolutely is. And it validates this whole complex multimodal approach and suggests you need to do everything at once, shock the virus, neutralize it, and boost the body's long-term memory. We're just waiting on the results from many more of these large combination trials.
So shifting gears a bit to the bigger picture, what about using bNAbs for prevention, for pre-WEP? It seems like the bar has been set almost impossibly high by long-acting RT.
The bar is nearly unattainable. You're right. Drugs like cabotegravir and lenacapavir are just phenomenally effective. A recent study showed lenacapavir, given every 6 months, was 100% effective at preventing HIV. How do you compete with 100%?
So when they tested a bNAb for prevention in the AMP trials, what happened?
The AMP trials used a bNAb called VRC01, and the overall results were disappointing. It didn't show significant protection. But when they dug into the data, they proved the mechanism worked. VRC01 was 75% effective, but only against strains of HIV that were actually sensitive to it.
Ah, so the breath didn't hold up in the real world.
Exactly. The big problem was pre-existing resistance. Only about 30% of the circulating viruses in the communities they studied were sensitive to VRC01. So for prevention, you'd need something with incredible breadth, like O4-AS6 or a cocktail of multiple antibodies.
Which, given the logistics of IV infusions and cold chains, just makes it a huge hurdle compared to a simple injection of lenacapavir.
It does. Future bNAb prevention trials now have to be designed as non-inferiority trials, which require massive sample sizes. It makes the future of passive antibody use for prevention very, very uncertain.
So if infusions are the problem, the future has to be getting the body to make the bNAbs itself.
That is the consensus. The main strategy being explored is genetic delivery, using something like an adeno-associated virus or AAV vector to deliver the gene for the antibody into our cells and turn them into little antibody factories.
And they've done this.
They have. In one monkey study, AAV delivery led to continuous antibody expression for up to 6 years. And in a phase 1 human trial, some participants had sustained antibody levels for up to 3 years.
But there was a roadblock there too, right? The body's own immune system fought back.
It did. In that human trial, 3 of the 8 participants developed anti-drug antibodies. Their bodies recognized the new antibody as foreign and mounted an immune response against it, which seemed to reduce its production over time.
So what's the ultimate holy grail then? The solution that avoids both the delivery problems and the anti-drug antibody problems.
Vaccine induction. Developing a vaccine that can actually teach our own B cells how to make these powerful, broadly neutralizing antibodies naturally. That would eliminate the need for any kind of passive infusion or viral vector. It's the ultimate goal, but it's also the most difficult scientific challenge.
This has all been about a functional cure, about durable remission. But we can't ignore that ultimate goal of a sterilizing cure completely eliminating the provirus.
It remains the ultimate prize. The stem cell cures showed it was biologically possible, but they also confirmed it's not clinically practical for most people because of the risk and cost.
Right, but now we have a tool like O4-AZO6, which seems to lock down a site on the virus that's almost unmutable.
And that's the interesting thing. If O4-AO6 proves as effective in humans as it was in mice, it could achieve a level of functional control that is so profound, so durable, that it gets incredibly close to feeling like a sterilizing cure in terms of effectiveness.
So, to wrap this all up, where does this leave us? The core challenge is still that hidden, latent reservoir, and testing new approaches still relies on those risky ATIs.
But the advances are stunning. BNAbs are incredibly promising, especially with the new engineering and discoveries like O4-AZO6. But the key takeaway from the clinical trials, from Titan, eClear roadmap, is that combinations is essential, but synergy is not guaranteed. Timing is everything.
We're really just at the beginning of seeing the results from these complex multimodal trials. The path to a cure is clearly intricate, and it's probably going to be highly personalized.
But the sheer technical advances, from the LS variants to the structural breakthrough of O4-AO6, it's breathtaking. We are closer than we've ever been.
Which raises an important final question for you, the listener, to think about. Given how hard it is to achieve that perfect, sustained immune response to clear every last infected cell, will the eventual breakthrough for a sterilizing cure come from a highly specific antibody like O4-AZO6 that just locks the virus in place forever? Or will it come from a technology still in its infancy, like CRISPR gene editing, that could one day surgically remove the virus from our very own DNA?
Both of those are on parallel tracks. And only time will tell which one provides the practical answer for the millions of people still living with HIV.
Thank you for joining us for the deep dive. We'll see you next time.